Good afternoon. So I'm Teresa Freitas. I'm a computer scientist here at Biotechnology University from the computer science department. And I will present a brief presentation about my last work, or the dream that I have now in hands. It's a bit, it's not in this main area, but it's about genetics and computations. So I think that I hope you will enjoy. So I will talk about a new vision for medicine using genomic technologies and using computational tools. And I will, during my talk, present a little bit of what I've been doing from these last 30 years, just for you to understand the way. So I can say that everything started when I left the island and joined the electronic department here at the Technical University of Lisbon, and my idea was to work on satellites. But I end up working on circuits, doing circuits. And this is a wafer. So what we have here is something like this. So this is a wafer with a number of electronic circuits, so chips, that you have inside your computer. So inside this wafer we have, for example, I think is 1,000 microprocessors, each one for one of your computers. So, and the goal of that, at that time, was to understand systems and to understand circuits. So when I finished my PhD, I decided to keep trying to understand systems, but now systems that I build not with silicon, but with DNA. And so I decided to go and to try to understand chips, but these chips are microarrays. They are chips where we have inside probes of DNA. It means that we have very short pieces of DNA that can find a complement in our genome. So, as I think you may know, that our genome is a double helix, so we have two strands, and if I try to break them apart, so one strand will try to find the other, the complement. So if I pick my genome and put my genome as a single strand inside of one of these chips, pieces of that DNA will try to find its complement. And I can use this kind of devices to do what we call genetic tests and try to associate those differences that I will find in our genomes to pathologies and try to do genetic testings. It's true that when we talk about trying to do genetic testing and use it in medicine, a lot of things arrive, and people think that we are going to try to find new ways to make human beings, nothing like that. So we are just trying to improve our lives, to understand ourselves, to understand our circuits, and at the end, to try to solve many problems and try people to live better. So, but these ethical issues can be raised. What about the genetic information? Who is the owner of the genetic information? What is the appropriate use of this data? What are the limitations of the genetic testings? What are the potential discrimination? But I may say that we should always look for the bright side of science and think that we can produce things that will be very useful for humankind. So, at the end, what we would like to do is personalize medicine, to understand our genome, to understand our difference, to understand how they shape our health, and how can we live better and longer, if possible, just to achieve the goals that Professor has just pointed in this presentation. So, and at the end, we would like also to have drugs with your name. It means to have drugs that will do only the good thing of the drug without side effects. So, my last project, and my last project means that on the last 10 years I was working in this project, we have developed, using a multidisciplinary team, we have developed a chip. So I'm back again to chips, but this is also a chip. So it's here, the chip. It's not of this size, it's much smaller, but we can do a number of things with it. So with this chip, I can test a number of pathologies. Now it is configured to test cardiac pathologies. And what we do, we have small probes of DNA. We can extract your genome from your blood, put your genome inside the chip, And if you have some kind of change in the genome that will lead to a specific genetic pathology, we will read a signal that will be read using a laser, and then we can identify what we call a mutation. And then we can associate the mutation to a specific pathology. These kind of devices, they gave us a huge amount of data. So together with these devices, we need to develop systems, computational systems, algorithms to do machine learning, to understand the signals, and to put together a lot of heterogeneous data, clinical data, genomic data, a huge number of data. So we also have developed clinical decision support systems that will help clinicians in the diagnosis and prognosis. So this is a very recent news in the newspaper that talks about the microchip and how the microchip can help prevent sudden cardiac death. I just took this example because this is always a very strong example. So I will show you some images of the result of this pathology. So if you are a very sensitive person, please close your eyes for 18 seconds, and then you can go back to the presentation. And I will talk about Mikos Fair. Mikos Fair was a football player from Benfica that was playing, and suddenly something happened. Okay, this is hard, it's quite difficult to see, and this is the result of sudden cardiac death. This is a pathology with a very high incidence in the human race, so 1 in each 500 persons can have a mutation for this pathology, but on athletes, high-performance athletes, this is a very problematic situation, because this is a normal heart, with a normal tissue. As you can see, all the cells are very well aligned, and the problem is when you do very sport or you are under stress, your heart can be like this, very sick, and the cells will be completely misarranged. If you have this mutation for this pathology, what will happen is that suddenly the electrical signal will stop, and the person will die. And we cannot cure this pathology, but we can prevent. And we can use genetic testing to do a number of preventions. And that is the best part of the story. So, due to this work, we just start a new company that is Art Genetics, and the company started today. So, it's a new company from a spin-off from IST, and as you can imagine, this company needs a multidisciplinary team. We have a PhD from Biotechnology here from Técnico, a PhD from Molecular Biology from Faculdade de Ciências, and myself from Computer Science here from Técnico. And what I think about the future, I think that the future is already here, is already there. So we already have devices that can travel inside our body that are of the size of our cells that can do drug delivering. We have this amazing system that is Watson from IBM that is doing machine learning using a huge amount of data, and that is able to help clinicians in the prognosis and diagnosis of cancer. So I think, and as you know also, you have a huge number of apps from your eye things that are able to measure your pressure, and in the future, very recently, I hope also to do clinical analysis. So I think that we are already living an amazing period of our history where biology and medicine and computer science will really make the difference and will play an important role. So, thank you.
Hello everyone, my name is Gabriel Pires. I am an associate researcher at the Institute for Systems and Robotics and also an assistant professor at the Electrical Engineering Department of the Polytechnic Institute of Tomás. I will give you a brief talk about non-invasive brain-computer interface, talking about some of the challenges of non-invasive BCI. Well, first of all, we need to define BCI. BCI translates brain activity that has been volitionally induced by the user into commands that can be used to control a computer or any other input device. So in a BCI, it is assumed that the commands are not conveyed using peripheral nerves or muscles. We have in this picture a generic view of a BCI system. First, we have the acquisition of the signals that can be invasively or non-invasively acquired. In the case of invasive recording, we have the intercortical recordings in the cortex. The microelectrodes are implanted in the cortex and records the spikes directly from the neurons. Then we have the electrocorticography. In this case, this is also invasive, but the electrodes have some millimeters and are placed in the dura mater. And then we have a non-invasive approach, which is the electroencephalographic technique, which records the signals from the surface of the scalp. This one is non-invasive. After signal acquisition, we have to process the signals with signal processing techniques, which have to extract the neurophysiologic features that are related with the neural mechanisms that are being used to control the BCI. After these features have to be learned with machine learning algorithms and classified and then translated into control commands. And these control commands can be used to control speller devices such as mental typewriters, mobility devices such as wheelchairs, games, prosthetic hands, or neurorehabilitation. There is always some sort of feedback, usually visual or auditory feedback, but also tactile and proprioceptive information through intercortical microstimulation or peripheral stimulation. So comparing my work with the work of Professor Kevin, I'm working in this, in this sense, acquiring information from the brain, and Professor Kevin is giving the information of feedback to the user. So it closes the loop. And this is a current research, a hot research topic. It is exactly to provide information about proprioceptive information. Well, comparing the three techniques, I will start by the first one, the intercortical recordings. Here, the signals are directly picked from the neurons, so it records the spikes of the neurons. It has a very high spatial resolution, a very high signal-to-noise ratio. And it is possible to have a very good accuracy about motor imagination. So it's possible that subjects control robotic arms with a very high accuracy. Some of the current projects, we can see here pictures of a project running at Brown University, the BrainGate 2, which shows impressive results of a woman controlling a robotic arm in a 3D environment. There are some problems with intercortical recordings, namely the long-term stability of the recordings. And obviously, this is a highly invasive approach, so there is risks for patients. Then we have also another invasive approach, but less invasive than the previous one, which is named electrocorticography. In this case, the electrodes pick the information from thousands of neurons, so it's field potentials, and the spatial resolution is about 1/10 of millimeters. The amplitudes of the signals are about 50 to 100 microvolts, and the frequency range is about 0 to 500 hertz. There is a low susceptibility to artifacts, so this is also an approach that gives good accuracy in controlling and in decoding motor imagination. And finally, we have the non-invasive approach, electroencephalography. There are several limitations with electroencephalography. First, it provides a very low spatial resolution. This means that if I have an electrode right here in my scalp, it will pick up the information not only under the electrode, but also in the region of about 10 centimeters or more. So the signal-to-noise ratio in terms of spatial resolution is very low. The signals are also very low, 10, 20 microvolts, and the frequency range is also very short, about 50 hertz. All this and the susceptibility to artifacts such as muscular artifacts, environment noise, all this make EEG a very noisy signal, so the signal-to-noise ratio is very, very low. But it's a non-invasive approach, it's cheap, it's portable, we can place electrodes all across the brain, so there are obviously a lot of advantages to use this approach. It's important to say that 80% of the research activity in BCI is with non-invasive approach. But in the last I would say the last 6-7 years, there has been a growth with intercortical and electrocorticographic research with quite impressive results. Well, who can benefit from a BCI and how? The main focus of BCI is the restoration of the communication for people who are in locked-in states. What is a locked-in state? A locked-in state means that the person is cognitively intact, but is unable to control any voluntary muscle. So it can, the person cannot speak, cannot move any muscle. In the classic locked-in state, sometimes there is movements of the vertical or horizontal ocular movement, but in complete locked-in states, the subject loses completely the ability to move any muscle. So this is the sole channel that a person in locked-in state can have. But BCI can also be used as an alternative communication for subjects that still retain some motor functions, but find difficult in controlling standard interfaces, such as eye tracking, head trackers, or head switches. So BCI can be another channel that can complement the other interfaces. BCI can be also used as a neurotherapy tool for children with attention deficit and hyperactivity disorder, can be used as a neurorehabilitation tool for people who suffered a brain stroke. There are several experiments that have shown that BCI can help to reinforce the neuroplasticity, which means that subjects can rewire the brain in order to perform tasks that were performed with the regions of the brain that were damaged. And finally, BCI can be used as entertainment or just for focus training, and obviously this can be used by everybody, including healthy and non-healthy subjects. Well, what are we doing in the last years with non-invasive BCIs? Well, we have made control, 1D and 2D control of cursor with motor imagination. We have developed mental typewriters, which means that we are able to spell letters sequentially. We have been developing games for children with attention deficits and hyperactivity disorders. Right now we are developing a game for children with neurofibromatosis. We have developed an interface specifically designed to steer a wheelchair. We have BCI integrated in ambient-assisted living scenarios and home automation scenarios. And until now, we have made some clinical tests with 14 subjects, 14 participants, 7 subjects with amyotrophic lateral sclerosis, 5 subjects with cerebral palsy, 1 subject with Duchenne muscular dystrophy, and 1 subject with spinal cord injury. I will show you briefly just 2 short videos. This is called the lateral single character speller. This combines 2 kinds of neural mechanisms. One is called the P300 neural event-related potential, which is a peak that occurs 300 milliseconds after a target event occurs. So the subject only needs to focus the letters that he wants to select, and this way, sequentially, letters are being detected. It is possible to have a transfer rate of up to 10, 12 symbols per minute. On average, from 60 persons, the average is about 5, 6 symbols per minute. If the user, if there is an error, the user can select the delete and spell the letter again. So this is a form of spelling with a relatively low transfer rate. I will show you one of the interfaces that was developed to steer a wheelchair. This is a human-machine shared control, which means that the user just selects the commands for direction, such as left, right, forward, destination goals, such as kitchen, and the trajectories are performed with the wheelchair, the navigation system of the wheelchair. So it's impossible to have a non-invasive brain-computer interface controlling in raw the trajectory of the wheelchair. But combining the information from the user with the sensing information of the wheelchair, it's possible to navigate the wheelchair safely. We are seeing the same experiment, but right now with a girl with cerebral palsy. She's not seated in the wheelchair for safety and ergonomic constraints. But she is controlling, the wheelchair is in the field of view, so she is controlling the wheelchair remotely. The commands are being sent by wireless, and she controls this interface quite efficiently, always with 100% accuracy. Just to give you just another example, the same girl controlling another interface. This one is the standard row column speller. I'm sure most of you have already seen this interface. She controls the interface with a transfer rate of about 4 or 5 symbols per minute, with an accuracy above 90%. We have tested with the 14 clinical patients. The severest case was the participant with Duchenne muscular dystrophy, because at this moment he loses completely the ability to control any voluntary muscle. And he controls also the BCI with 4 or 5 commands per minute. You'll see just in a moment. This is the participant with Duchenne muscular dystrophy, which controls the BCI with 4 or 5 symbols per minute. Okay, what are the BCI challenges, main issues? Well, as you have seen, BCI has a very low transfer rate comparing to other interfaces. But we have to see this point relatively with the degree of disability of the user. But in relative terms, BCI has low communication transfer rates and a lower reliability. Due to EEG non-stationarity and external changes, there is the requirement of a calibration session before the user controls the BCI. I have reduced the time for calibration to, in some of the interfaces, to about 2 minutes, and in the other interfaces to about 5 minutes. This is quite good in terms of state of the art. But there are other problems. The non-stationarity of the EEG signals makes also the user performance decay during the session, because the models that are decoding the brain patterns does not accompany the variation of the EEG. Then we have some usability issues, such as most BCIs are not self-paced by users. What does this mean? It means that most of the BCIs are always translating the EEG into commands. So if the user in a given moment doesn't want to provide a command, the processing and machine learning algorithms have to detect that the user doesn't want to convey a command. At this moment, I have already non-control state detection with a 95% accuracy, but this is not a solved question. Then we have also the problem of switch on and switch off the system. Users cannot switch on the system and off. It is needed new mechanisms to switch on and off the system. Then BCI requires more focus and training than other interfaces. So it is much more tiresome than other interfaces. And final, but the most important, electrodes take time to set up and are not natural. So the electrode setup is quite cumbersome in most cases. How to move toward BCI as a practical assistive device? Well, we need to develop hybrid systems that combine different neurophysiologic mechanisms, which means that we have to combine motor imagery with event-related potentials and any other neurophysiologic features. This is the first step to improve the transfer rate and improve the reliability of the BCI. And then we have to think an assistive device where BCI is just a part of a multimodal interface, where other sensorial inputs are also part of that multimodal interface. So you have to think in order to have a complementarity between several sensorial inputs. We need to propose new and develop new adaptive and unsupervised signal processing and machine learning algorithms to cope with the non-stationarity of EEG signals. We need to detect non-control states and mental states, such as fatigue, workload, frustration, because it gives important information to adjust the algorithms. We need to develop new control designs relying on human-machine shared control and human-computer shared control, and have new human-computer interaction approaches. And finally, we need to develop dry electrodes with signal-to-noise ratio similar to the wet electrodes. So we need new electrodes, new biosensors, because I think this will be the main step to improve a BCI and to bring BCI at consumers' home. Okay, thank you.
Okay, my name is Moisés Piedade. I'm retired from IST and work in research in SKD. In last years we have worked in micro bioelectronic systems, mainly in bioships, lab-on-chip and lab-on-pocket, and electrical stimulation, neuron electrical stimulation. And also now you have a project in non-invasive neuronal stimulation and sensing. in the area of bioships, we are using a technology that was developed by INESC MN that is based on magnetic sensors. INESC MN is a world leader in magnetic sensors, and they develop very high sensitive magnetic sensors, and also several applications in several areas as non-destructive testing, biological systems. And the basic principle here is that we use magnetic spheres, very small, 25 nanometers or 250 nanometers. These magnetic spheres are prepared with biological solutions that attract organic material, and we carry these magnetic spheres over a circuit where we place probes. Biological, for example, DNA complementaire of one that we can analyze, and we force these magnetic particles to travel over the ship, and if there is a correlation between the DNA and DNA complementary of these materials, they aggregate, and after this we wash the ship, and some of the material, magnetic spheres, are placed over the sensors. They are not wiped out, and we can read this by electronics means, reads the magnetic spheres that are over the magnetic sensors. We also developed a system to do that. Nowadays we have a lab-on-pocket system. This system can do complete clinical analysis of several types and DNA analysis at this moment, and you are trying to push this to a small ship, a very small ship, in order to get the lab-on-ship system. This project was in this project collaborate several institutes, INESC MN, INESC ID, we developed the microelectronics and the signal processing and interface, computer interface, IBB, surface biochemics, microfluids. We have microfluids over the ship, microchannels where we pump fluids over the ships, and we substitute all this equipment by this one in this lab-on-pocket system. Just small reference to the system. This is based on digital signal processing. We have embedded system with pump controllers, current drives for reading the magnetic sensors, and we have also a model for communications on a cryptographic engine, parser, and communication manager. There are several work here in the communications area. This was our first array with about 256 sensors. And this is the model. It's a very small model compared with our card. So there are some in this video. Okay. We can present a technology that was developed by INESC MN that is a way to transfer all the fluids. Sorry, because the video is not running. Yes, now it is. We apply an alternate current of a very small frequency, and the particles are attracted to this loop. We place the sensors inside this loop so we can put all the organic material over the magnetic sensor. By this way, all the spheres, magnetic spheres, are carried over the sensor. So, these are electronic circuits that we are using in this. We have also a biodatabase portal with this system. Now we are developing, we developed a technology that is based on CMOS, conventional microelectronic circuits, and it was possible to integrate these nanosensors, very high sensitive sensors, with thin film technology over the CMOS circuits. We have registered this patent international, and now we have what we know, what we call the biomagnetic CMOS biochip. So we can do all these operations over a small CMOS circuit. This was our first prototype with 16 sensors. Now we have a race with 256 sensors, and we are working for 1,000 and 2,000 sensors over a small CMOS circuit. And this small circuit contains all the electronics to drive and read the sensors, to do the small signal amplification with ultra-low noise, and provides an analog signal in a very high level that we can amplify and digitize on the processor. Now we have we are world leaders in the performance of this system. We now have a system with 32 bits and architecture based on 2 ADCs with 32 bits and 24 bits in parallel. And we have developed algorithms of signal processing in order to increase the resolution of this system. Now we have a noise lowered to 4 nanovolts per square root. In the last platform, we have 70 nanovolts. And now we are working in the power consumption reduction. So, in a few years, we don't need a doctor we will not need a doctor to solve all the problems. Now I have some slides about the cortical interfaces. As you know, electricity can die. This is a small video, just Edison invents the electric chair, and also proved that AC current is more dangerous than DC, because Edison works on electrifying cities with DC current and Tesla with AC. So the worst video is not working. Why not? Okay. So there is Edison works in cinema in 1930 and registered the first films killing elephant. But Edison was not sure that he was able to die the elephant, so he put some cyanide in some elements, and okay, electricity may die the people. Now we have developed some work on EEG. Our idea is this was fully developed in IST, a system based on microelectrodes and small signal amplification and an FPGA concentrator. This system works with about 128 electrodes. Our idea is to develop a system, an helmet with all the contacts, all the electrodes, and to communicate wirelessly the system. So we have to concentrate this in FPGA. Also, now we are developing microcircuits for small arrays of 16 electrodes in order to concentrate the signals and to provide this to the FPGA. We are working with several implants, microelectronics for stimulation and also for sensing. One we have worked several years is the vision, artificial vision, with implants for the artificial vision. This is the first implant done by the Dobel 20 years ago. We are collaborating with Professor Lobantunes. Professor Lobantunes was the most implants, the large number of implants. They were done in Lisbon, not in the United States, where the laboratories are located. This is the implant that was retired from a patient here in Lisbon. This technology is very old, and this is not efficient for several. They have several problems. So what we are working is a combination of new type of microelectronics with microelectronic circuits connected directly to the microelectrodes. But this needs wireless energy for these implants, these circuits, and also we have to develop ultra-low power consumption protection circuits. Also, we are working in the robust communication systems using technologies based in ultra-wide bandwidth for this kind of communication. So in Cortivis and ICONS projects, we developed a system. Basically, if you know, this is image capture, visual encoding, and after this is a radio frequency modem, and we have a stimulator, in this case, UTA microelectrodes array, and we have developed all these in chips. So here is a surface microstimulation that was, this type of stimulation was used by Dobel in the first visual process. We used the UTA microarray because it is proven that we need to penetrate about 1.4 millimeters in order to stimulate the visual cortex. These are typical values of current stimulation. We needed a very, with surface stimulation, we need very high currents, and problems of epilepsy may occur. Okay, this system was. We have an image. We process we partition those are partitioned of images in small blocks, and we filter, and this is the reconstructed image that is transmitted by the system. There are circuits, electronic circuits that are used for this. There is a problem with these implants because cortex is not easily penetrated, so Professor Eduardo Hernandes de Alicante developed the system based on a pump system, pneumatic system, in order to get an force impulse to penetrate. This is the microelectrodes that we use in first experiments. We done experiments on rats, cat retinas, where we develop, collecting the signals obtained by the cat retinas, we can process and model this behavior, and we do electronic circuits that simulate this behavior. We develop CMOS circuits for the transceiver, radio frequency transceiver, and power transceiver we have a unit outside the head and a unit inside the head. We have to communicate and transfer energy from outside to inside. Okay. In the electrical engineering department, we have a demonstrator we call Elonica. Elonica has one of these systems and several gadgets also for demonstrating several principles used in electrical engineering systems. So if you use this image, this is the original image, decimated, because we have only 1,000 of my electrodes, and we have 1,000 of pixels in this image. Now we are working with doctors in developing microelectrodes for Parkinson deficiency. And this is an area of deep brain stimulation. Deep brain, about 10 centimeters of penetration of free electrodes inside our head. And in a small region. These microelectrodes may need to be placed in a small region in order to disturb neuronal system that has this dimension of 8 by 3 millimeters. So we have to put a stimulator, electrical stimulator, that generates electric fields that masks this neuronal system. I can put you, this was recorded from a patient that was treated with this. This operation is very expensive, about 30,000 euros, and only patients with very high F disorder are selected for this kind of operations. The stimulator. Now the doctor is programming the stimulator. I am authorized to show this video, but the results are spectacular in the same case. So, but this stimulator needs to work 24 hours per day, and stimulation is about 3 to 5 milliamps in the electrodes, and we need big batteries in this stimulator that is inside the body of the patient. Now, what we are working now is on magnetic detection without microelectricity. Instead of the registering potentials, we need magnetic. We register magnetic fields, and registering magnetic fields, we don't have physical electrical contact between the body and the metal, and we have not potentials and corrosion of the microelectrodes. We are working on magnetic stimulation, micromagnetic stimulation, and micromagnetic sensing using the high sensitivity of the sensors produced by Inés. The tests are done with pig because they can induce the Parkinson by chemical drugs, and we can. I don't translate this for English because we use the head of the pig without the body. Okay, just for our students, to work in these areas is necessary a very interdisciplinary knowledge. So, thank you very much.
Okay, thank you, Professor. I will now start to ask a question to the panel, picking up on some things that were said during the presentations. Being this a very multidisciplinary area, what would you recommend, or what would you think that could bring students more, well, closer to this area, could bring them to do research in this area? What would you think the requisites or the prerequisites would be for them? What would you look in a student for a researcher? And I would start probably with you, Cameron. You've been speaking in a long time, so what would you think about this?
Yeah, I think it is an area that involves humans, it involves technology, it involves medicine, and I think with humans and medicine, traditionally, it's been more of a biological understanding that people have. I think one thing to realize here, I feel all of us are perhaps more technological people, electronics, computing, and this is something, bringing this to the area, it's refreshing in itself. The human body is electrochemical, it's mechanical, and even looking at how the human body, in particular how the human brain operates, you need more, I would say, skills in terms of electrotechnical computing skills to understand what's going on in a fresh way. So I think you do need to allow for the inputs from the biologists and medical people and to be able to take those on board and adapt appropriately. But I think there's a lot of room for electrotechnical computing people to get involved in the area. Well, you don't have to be scared if you don't understand too much about the biology. You can't know everything. And I think a fresh look at how the body works now because of this technology is having to take place.
Not yet? Okay, okay, thank you. I would now ask Professor Anna, in more of a national perspective, and of the EST in particular, what would students need to do to follow these areas, or to collaborate in these areas?
Okay, I think, for example, since you are computer scientists, you have the opportunity to choose a number of courses that you will give you the basis. So in your course, you have courses like elementary courses on biology. You can choose them here at the university. You have elementary courses on computation biology, and your background is computer science. So as Kevin has said, so we need your expertise, and you need your open minds just to help these teams, because at the end it will be a joint work. But I think that here at this university you have all the courses that you need. You don't need to go away. So perhaps you need some courses if you want to know more about physiology, to go to the medical school to understand a little bit. But you can choose, because you have optional courses, so you can choose. So I think you have everything around you.
Okay, thank you. I don't know if the audience has some of... Okay, there's a question.
Okay, so my question is mostly about... we're seeing nowadays a lot of new courses, like, for example, biomedical engineering, that I think the IST course has 15 years, if I'm not mistaken, or 12 years, something like that. So what is the relevance of these courses if you are all, your basic is like computer science and electrical engineering? Is there a difference? Is it important to have this broader view on the subject before working, for example, in your projects?
Okay, I know the biomedical course very well, since I'm the representative from computer science in that course. It's quite different from your background. So they are not computer scientists. Oh, you are biomedical. So you are not a computer scientist. You have a very broad view of the area if you want to work close to medicine. That's something that computer scientists doesn't. Okay. I think that you are very well tailored to work in specific problems, for example, like these ones, problems that put together medicine and electronics and signal processing. But for example, if I want you to build a system or to design an algorithm, you will be able to design the algorithm. But if I want you to implement that algorithm to deal with 5 terabytes of data, I need a computer scientist, okay? So the point is, for example, I have a group on computational biology at INSKD, and I have computer scientists, students from biomedical course, students from maths. We need a lot of people from maths, even to deal with biostatistics or to deal with the data structures and even the algorithms. To deal with strings, that is something that they are very good. I think that you all share pieces of your pieces of knowledge, but for this kind of works, we need the whole team together.
Can I just add, I mean, I don't know the specifics of the course here, but biomedical engineering, for example, courses vary enormously around the world. And I think you really do have to look, this is a subject area, you do have to look at the course content very closely. Some of them can be very bio, and you know, and it's the same with biotechnology. Biotechnology can mean something that's very biological, but it can mean something that is very technological. For me, and I guess for the people here, we are more on the technology side, but it's important to look at the biological side to some extent. But I think you do have to be careful. It can be a biology degree that is repackaged, and that is a bit of a worry. I'm not saying that's the case here at all. In fact, I suspect it's quite the opposite, but that has to be a worry sometimes.
Here at the university, it's a course with a very strong background in electronics and in physics, so that's the basis of the course. But we are changing a little bit the course. You will have new directions from, I hope, next year, because now it's just an integrated master, and we hope that you will have different areas after in the master part, because you can work on signal processing, or you can work, for example, on clinical decision support systems. It's a completely different person to work on both teams, or biomechanics. Okay.
Okay, thank you. I would now. You were like, okay, for
something. Well, I have already supervised some students from biomedical engineering from Coimbra, from the course of Coimbra, and effectively, I think that specifically the course in Coimbra has too many areas. So the students don't focus anything in particular. So they know a little bit about many things, but nothing about a specific topic. But in the other way, last week I was invited to a workshop from the medicine faculty from Coimbra, and the audience was only students from medicine. It was my first time for such audience. So it was difficult for me because I didn't know if they would understand my language. And at the final, my impression was that effectively we were not speaking the same language, and there is a big gap between medicine and electrical engineering. So maybe biomedical engineering can do the bridge between these two scientific areas. But the most important is to, in research centers, to have people from every research fields. So this is the most important, and I think it is not always, in most, well, from my experience, it's not easy sometimes to join people from different and very different research scientific areas. And I think this should be the effort, and this can be done in the research centers.
Okay, I would now launch a question more about, would you like? Okay.
Just to confirm that to work in these projects, a very interdisciplinary knowledge is necessary. So, but I think that it's best to do a course with the things are rightly put and studied, and after this we can get courses on specialization in different areas, advanced specialization, but you have to, in some subject, you need to be very well prepared. So my opinion is that it's best to have a good foundations in a small area, and an open mind to absorb knowledge from different areas. In our case, we advance very much when we put electronics with physics work together. They develop several things very good, but these things are belong to electrotechnics and physics. So we have, in this case, I am very satisfied with the results that we achieved, because myself, we cannot get these results without they, and vice versa. Okay, so everybody is satisfied with the results, and I think that is the best way to go to these new areas. But my opinion is that all the systems are more and more interdisciplinary. So we have to in our course in the IST, we have to have a very broad area of knowledge in courses, but some spinal structures of the course where we are deeply we have a very strong courses in some spinal course, but we have to put options in order to students got different knowledge from other areas. Okay. But I would prefer courses based on a spinal structure with different apertures to different options. It's my opinion.
Okay, I don't know if anyone at the audience has a question. If not, I would like to move to a different topic. What I would like to ask, when you, in all of your presentations, you mentioned something where you show like to compare a lot the human body to a system, and you treat it like it. How dangerous do you think that comparison might be? And I would send it to the panel. I don't know if all of you want to answer, if only one of you. No, I would start with...
Who wants to answer?
Well, I don't think it's dangerous. I think we need to look at the human body as a system much more. We need to understand it much more in a technological way. For example, and I think it gives us a different understanding to say how a medical person would look at it. For example, if I take the human nervous system you can study and model as a resistor-capacitor network, and you look at In terms of signals moving spatially and temporally, you can model how long it's going to take signal very accurately. Sometimes the models are more accurate than, say, the Hodgkin-Huxley models in the neurological sense in terms of times. But in doing that, you realize that the human nervous system is an antenna. It's like an aerial. It picks up signals. Now that means something. And it means that our nervous systems pick up all sorts of signals. And hence, when somebody says, oh, cell phones don't affect our brains or our nerve, they're perfectly safe, well, technologically, well, hold on a minute. You can't exactly say that. They do affect. Maybe we don't know how they're affecting it. We need to study more. But we can, well, it does affect the nervous system. The signals pick it up. So I think, far from being dangerous, I think it's dangerous not to look at the body in a technological way. And it gives us another insight when we look at it in a technological way, which, as was being pointed out, in the medical world, they do tend to talk a completely different language and look at things in a different way. And I think it's important to look at things the way we do. The systems way is a necessary way to look at the body.
I would just like to add something about what Kevin said. And how accurate do you feel the models of the human body are right now? What do you think they bring to the area? How do you think they will tend to evolve?
Okay, they will evolve a lot. And in some points we are very far away from what we really are. But I agree that we should look to the body as a system. And I think that when we look like that, we became more human, because we don't give space to speculations. We know that things are what they are, and perhaps our emotions, they are just the result of a bunch of drugs that are misbalanced in our body. So we don't, perhaps we are becoming more human in a sense, in a more psychological sense, okay? So, but I like to see us as a system.
I don't know if anyone from the panel wants to answer.
About the models, well,
Okay.
The models are very coarse, very simplistic. So obviously we need to evolve a lot about modeling brain and other systems. I know that the European Union is spending, I think, 1 billion euros to have 10 billion.
It's 1 billion, so it's more than 10 million.
Yes,
so
1
Gabriel
billion
was asking for 10 million.
to build a complete map of the brain. So obviously our knowledge about the brain is still very coarse.
Yes, the system is quite complex, and it's good because you have a lot of work to do. But in a number of things, it's also nice to see that you already have some answers, so we can have correlations and solve problems and give some results. So this is the way.
I don't know if anyone from the audience has a question. Right there.
Hi. I want to ask to Kevin and the others too, if he, in his personal experiment, if he was able to produce a signal without moving the hand in the chip he used, and for the other ones, with the EEG, with the external sensors on the head, if it's possible by thinking to produce a signal in a singular electrode or something like that, or if you only capture a pattern in all of the sensors.
Okay, to answer you straight away, no, I had to, it was motor signals that we were picking up. So and we were picking them up from the peripheral nervous system, so I had to move my hand in order to generate the signals. The other gentleman showed the picture of the brain gate being used in Brown University by Lee Hochberg, John Donoghue. I mean, there they've got patients who are paralysed, that can't move, but they still generate the same signals in the brain by thinking about moving. So they can think about moving their hand. So it was exactly the same signals we were picking up with me, but because I am, my hands operate, so we pick up the signals lower down in the body, which again is a lot much safer than going into the cortex. But I think as well, if we'd done the experiments and I said, hey, look, the light's coming on and I wasn't moving, people would say, this is just some trick, you're just switching the light. So in a way, to show I move the hand and then the light comes on, but that's how we had to do it. But it was picking up the same signals as they're now using in Brown, but picking them up from the cortex. So.
Can I answer? Yeah. With non-invasive BCI, with EEG, it is possible to decode several patterns associated with motor imagery. To make 1D control and 2D control of a cursor on a computer screen with detection of 3-4 patterns. Usually the imagination of the motor activity is something like, imagine that you are closing your hand, your right hand, your left hand. Imagine that you are moving your feet, and imagine that you are moving your tongue. These are the four typical brain imagery that we can decode. The problem is, comparing to invasive BCI, is that the spatial resolution is very low. So if you imagine here your motor cortex and your sensory motor cortex, the problem is, for instance, here you have the part that controls your foot, and here you have the part that controls your hand. And when you are doing this, just in the top, you are also picking the information from the activity that is being generated in the region of the hand. So you have to apply robust signal processing techniques, usually spatial signal processing techniques, and with this you can improve significantly the signal-to-noise ratio. And yes, some of the experiments I didn't show here, but some of the experiments we have already made, some people playing Tetris with motor imagination simply by thinking left hand and right hand and moving the piece to the left and to the right. This is 1D control, but also 2D control is possible. There are some experiments showing 3D control, but this is much more difficult. But yes, there are some experiments with motor imagination that are that allow to do something close, but to what is done in invasive BCI. Okay.
No more questions? Okay, I would like now to, no more answers from the panel? Okay. I would like now to introduce, again, another different topic, a little more vague and maybe more tough, in which I would ask, if you compare the human body to a machine, what kind of, and being all of you with various of different technological backgrounds though, what kind of ethical questions would this bring? So when you compare totally the human body with a machine and you model it and all of it, what really makes us different from a machine?
I mean, really what we are as humans is our brain. The body nowadays, to be honest, it's obsolescent. We can see we have problems in the Western world with our bodies now. Particularly in the United States, the number of people that are obese to have to, because the world has changed. It's a very different world to one even 100 years ago, where people were required to do a lot of physical work. Now, because of the way technology is, and we travel most of the time by sitting down, with our bodies are not really suitable to the world we live in. It's just really our brains. And I think a lot of people maybe will take 1 hour or 2 hours of physical exercise every day just to keep their body. What a waste of time. What a waste of time. Wouldn't it be better if we could use that time for doing other things? So I think from an ethical point of view, the sooner we can get rid of our physical bodies, the better, and use all sorts of pieces of technology. I mean, at the same time, often people die because they have a cancer or some other problem in their physical body. Well, what a shame. Your brain is highly active and can go on for many more years, and that's the end of it because some stupid thing with your leg, that why bother with this part of the body? So I think the sooner we can get rid of the physical body and put a robot body or something that we can keep our brains housed in. The brain is the important thing, and keeping that going for a long, long time, that's the critical area. I'm not sure that that's answered the question, but that's my thoughts on it anyway.
Yes, it was a very strong answer. Strong but very interesting, because the point is that Kevin, you want to keep the brain, but the brain is built on a DNA-based platform. So I think that we should get rid of DNA-based platform at all. So, and go and move to silicon and map our brains in silicon and keep just the computers, okay? So, and then you can go farther. Okay, yes, so we remove. This is just a way of thinking. Should we choose a silicon-based or a DNA-based platform? So what we see is that the DNA-based is quite robust, and the silicon one is not yet accurate enough to keep our DNA-based. But at the end, we'll get there, and I believe on that. Okay, but what about getting rid of the body and keeping the brain? I think that we are facing now a very interesting thing, that is, we are keeping the body, and if you can see around us, we can have 80 years with a more or less nice body and a complete a brain that is a mess. So people, this is the problem with the retirements and with the payments and the toll, because people are living forever, and just we don't have enough money in the world to keep like this. So the bodies are are okay, and the brains are not. So people need to stop working with a good body. So, but we cannot compare our body. We don't have a system built in silicon that can be compared with the complexity that we have at the moment in a DNA base. Okay? But I think and I believe that in the future we can map everything in a different platform and go. Okay? But we have a very long history with DNA, so and the world is DNA, and our cells are around. I don't know.
Okay, thank you. So any other member of the panel wants to answer it? Okay. So any question from the audience? On a following up question, I would part it in two parts. Do you really think that we'll get there anytime, even in the long term, to that point when we can simply get rid of the body and just use the brain, put it in an artificial body or container, even if it is, and just move along? How far are we from it, and what are our, well, let's say, obstacles besides money? Besides money.
Okay, I think that we are a bit far. And I think the main obstacle comes from the fact that we are a machine that are trying to understand the machine. And I don't know if this machine can understand the machine. Okay? So it's quite simple for us to understand other machines that are not us. Okay? And I don't know if at the end we will have that many obstacle that is a limitation of this machine to understand himself. Because it will be nice to map everything in a... I think that we'll still have a number of obstacles, but I think that we are really living in a period where we will achieve many, many, many new things. So we have the data. Things are... You can think, for example, that you can build algorithms that will learn all the events, and then you just remove the brain and put a chip. And perhaps the chip will answer. And so you can use the Turing test to understand if it's a computer or not. Okay?
I would like to think it might be possible in the future. There's a wonderful story by Franz Kafka called Metamorphosis. If you haven't read it, read it. It's a wonderful story. From a technological point of view, it's quite inspirational. I won't spoil it for people if you haven't read it. Just sit down and read it. I think with, you know, when you, the sort of stuff we're doing with brains and putting them in robot bodies, I mean, it would be possible if there is a volunteer now that we take the brain cells fresh from them. Okay, maybe they die as a result, but that's it's very good for them to volunteer. And we then put them in a robot body. I know certainly Thomas Berger in USC is doing research on taking slices from brains. So as long as we could take slices of brains and put them in a robot body, so partially we could do what you're saying now, not as a whole brain, but as part of the brain. The question then would be, would any of the original content or power, a memory perhaps, or anything, or knowledge of how to do something, would that be retained in the little robot body? Don't know. That would be. We need to try it. We need to find out. I mean, maybe if what you really wanted to say was to volunteer your brain for the experiment, then let's go and do it.
Okay, but I can add something that is, okay, this is a nice view that is to put the brain in a robot body. But we can think, for example, about the Google glasses. Okay, you know, you heard about the Google glasses, that you will see all the information. And if you put a number of devices around you, like the Google glasses, suppose that the Google glasses give you all the information and can make all the decisions. You just ask for something and he runs an algorithm and he gives you the answer. So you are putting your brain outside you. The brain is already outside you. Perhaps in a number of years we will have just a controller, a very small controller, a DNA-based controller that is controlling devices that are around us.
Okay.
You don't need to put the brain outside.
That probably picks up on the second part of the question, which is, I believe Kevin spoke of the brain as a centerpiece of everything, and I would ask, I find the brain pretty limited itself, so why not replace it too? Why not enhance it? Is this possible? Is this completely insane? Can we get rid of it?
I think certainly, I don't know that I do see it as a standalone. I think it's important is if it like computers, you know, you can have a computer that is on its own, but the real power comes when you network the computer, and you can share knowledge and the intelligence is shared. I think that's true with the human brain. At the moment, this is as close as we get to networking realistically, which is not very close at all. The possibility of networking the human brain by using implants is a realistic one, and that's where the benefits come from. As for how important then, we're into Star Trek, I guess. It's your brain is then a node on the network, which has much more important and it's much more capable, it's much more powerful because it is part of the network, but how much of an important node is it? I would like to think that, you know, my own brain still has some importance as a node, but maybe that's just wishful thinking.
Right there.
Professor Ana Freitas was saying that you in Google Glasses, for example, you have to ask for something and you get the answer. Just idealistically speaking, it would be something like that. When we will reach the moment when we don't need to ask for something, we just need to, for example, oh, how many hair is in my arm, and I will know the answer because my eyes or whatever is replacing it will just do the math for me. What level can we get of these kind of answers? I just mean, like, okay, how many hairs in my arm is just it's feasible, but some limitations. I don't know how much of an overflow can we. Where can we get the answers? What is the limit of the answers that we can get from this? Like, can we just enter a loop or something?
I don't know if you will have a limit. You can imagine that you can lose completely your memory. You don't need to read anything. You don't need to memorize anything because you will have everything in the cloud. Okay? So you just need to go to the cloud. And that's what we are already doing. I think that without the internet, if someone asks you something, or you know, or you have read,
just Google
or you
it.
just say, I don't know. Nowadays, no one says, I don't know. Just a minute, just go to Google. Okay? So everybody knows everything. So our memory is already outside. There's a number of questions that are already outside us.
It's become a collective memory, which is the point. You know, again, it's a network memory, and there's no problem with outsourcing your memory. Is a good, why not? I mean, we know our memories are not very good. The important thing is still, though, to be able to recall memories. In this case, how to access and how to get the information becomes a more important point. But I think it is something, outsource your memory, as to an extent we're doing, is a very sensible way to go, which frees up part of your brain for doing other things, whatever that happens to be.
I believe the professor.
I think the problem is not a problem of memory, it's a problem of intelligence. And we have everything around us, but we have a different machine that is a very clever machine, the most intelligent machine that you have, someone conceived. But I believe that is possible with the technology to improve or to interact directly with our head. Okay, directly, with microelectrodes sensing signals and correct the signals in order to get different actions from a head that is not working very well. Okay, so I think there's a very Professor Raul from MIT in Biotechnologies this year present a system that the human body is in a feedback loop with machines, and there is some perspectives to control some functions, integrating different intelligence inside our head in order to correct some functions that are not working properly. I give you a very simple example that is Parkinson correction, but there is some things. For example, if you are sleeping, you can see images, but the images are not entering by your eyes. Okay, these images are created inside this machine. So I think that is possible to induce these images directly without the visual system. So we can change the information generated by Cortex, inducing some machines. Actually, there are some signals that are synthesized and put in next May or June. I will participate in a project like this one, where we collect signals from a monkey. We process the signals and we put new signals in the microelectrodes, and the monkey changed the function that was predestined to be done. Okay, so it is possible to integrate machines in feedback with the human system.
Just want to add something. Well, neuroplasticity is quite impressive. Some years ago, or some decades ago, people think that the brain was completely still and without ability to learn new things. And we know that people who suffered from strokes and who lose the ability to control some part of the body, the speech, can rewire the map, the brain maps, and acquire the ability to perform tasks that were developed in other region of the brain. So I think these two concepts, one concept where our brain is supported by many intelligence. I like more to see the opposite. See the things that can improve our brain to learn more. I think this is more important than to have our brain receiving stimuli and supported by intelligent systems that are surrounding and are receiving information from the world. So we can do that, but we can also establish mechanisms to improve the ability to learn more and to adapt more to new things. It's not the idea that we can have a contribution to the evolution. The evolution is a very slow. Is very slow. So, but we can take more advantage of our brain. And I think it's important to see this aspect, because we can learn during all our life. You can learn new things, and we can do things that after training we can do new things. So I think it's possible to have new techniques to improve our learning capabilities. I think it's neuroplasticity. It's amazing in our brain. And there are some tools that can reinforce and accelerate the process of neuroplasticity. So I think these are several concepts of the whole system.
Okay. So I want to talk exactly about neuroplasticity, because I was thinking that we're not talking about substituting the brain or having some other medium to make decisions, but it's interesting how many great groundbreaking discoveries have been made during sleep, when people are asleep and suddenly they wake up and they know the answer to what their problem was. And that happens basically because of neuroplasticity. It's during the sleep that our brain makes new connections between the neurons. So to what extent would it be ideal to, okay, our brain is asleep and we have some machine that is detecting what he's doing and giving the answers? And another question would be about, for example, fMRI, because there are some studies where they now can decode which is basically a big coil, the fMRI, the electrical mapping of the images people are seeing, and probably they can decode the mapping of images people are dreaming or just thinking of. So if you can decode, you can probably input that stimuli to the people. So, what?
Well, I saw some videos of these experiments that you are talking about, and they were quite impressive. But this is quite is biomimetism, in the sense that you are seeing something And now you are recording the activity with fMRI, and then what was done was to reproduce, to collect a large database, and then to make the inverse process. Well, it is not exactly decoding the brain activity. It was more than recording a large database that corresponded to the images, and then do something like the inverse process. And when someone was thinking or imagining one of the scenes that were seen, you just picked the one image from fMRI from your large database. Well, I don't call this exactly the coding of the brain activity, but obviously you can have information from this mapping between the input, the sensorial input, and what is passing what is happening in your brain. It can be used to make algorithms for brain decoding, but this is not exactly brain decoding. So I don't know if it is possible to have something that captures your dreams. Well, I don't know if it's possible. I don't think so. Right now, at this moment, I don't think it's possible.
But that will be the dream of students, yes? You just need to sleep.
Okay, does the audience has another question? Nope. Okay. We're pretty much on time, so I'll just end up with some quick kind of cliche questions. So how far are we to technology can cure some of the biggest monsters as diseases like cancer and all of that? And we pretty much wrap it up as we are a little past time.
All the students that came to me to work on computational biology, they want to solve the problem of the cancer. Okay, so I think that we have a huge number of students that want to work on that. The problem is that there are a number of cancers that we can already solve it, and that you have very nice answers to the problems. But what we can see is that the cancers have very different ways and of progression, and are quite different. So, for example, when we talk about the breast cancer, that is a quite difficult cancer, because as more as we know, we have so many different aspects of that cancer, so we need to characterize the person. But I think that for many pathologies, we already have very nice answers. And sometimes we cannot solve it, but we can prevent, and that is already a very big step.
Thank you. Does anyone want to comment on this? Okay. I believe my colleague here has another one. He would like to ask very much. So in the meantime, I'll prepare the finals. He'll do it.
So hello, greetings. We are talking about previously for extension memory, but can we already interpret human memory? How permanent it is? Do we know how does it work actually?
I think we know very little. The point made there of sort of looking at visualization is about as good as we can get. But I think with some memory problems like dementia, Alzheimer's disease, we don't really know exactly what the problem is. Is it a problem of the memory storage? Is it a problem of the route to recalling memory? Is it a problem to the actual process of recalling memory? We don't really know what the problem is. So I think it's not just retaining memories and perhaps being able to plot them externally, but it's also a case, which we don't really know much of anything about, but it's a case also of how do you get hold of them and how do you then play them or relay them. So there's an awful lot we don't know. What we do know is it is electrochemical storage at a particular time in the brain, and we know where the brain is. So there has to be an answer that we can find out, but we're just quite a long way away from finding it out at the moment.
Oh, thanks. Does anyone have a question?
Okay, so I would like to thank all our speakers and thank the audience. Thank you very much. We'll now proceed to... I'll now switch to Portuguese. Sorry, Kevin. Vamos proceder ao sorteio. Confirmem apenas se ninguém tem um número superior ao 67. And I would like to end up with a round of applause for all our speakers.